Hello, and welcome to the next presentation in a series of presentations on adipose technologies. In this presentation, I'll be talking about the history of methodologies used for the isolation of cells from fat, and I'll also be discussing the nomenclature that has evolved during these studies. Some of the first studies describing the isolation of cells from tissue were performed by an investigator by the name of Alexis Carrel. In this publication in 1912, he showed that you could take a piece of connective tissue, which includes fat, isolate that tissue, and then mince it heavily, wash it, centrifuge it, and you would end up with a pellet of isolated cells as well as tissue. Carrel went on to show that these isolated cells in this tissue could be placed into tissue culture and individual cells would proliferate. This is probably the first example of what is now known as non-enzymatic cell isolation from tissues. Now, while not specifically describing fat-derived cells, this was a generic publication on the isolation of cells from a number of tissues, embryos, as well as minced connective tissue. The first enzymatic cell isolation from fat was reported by Martin Rodbell. Marty Rodbell was an investigator at the NIH, National Institutes of Health in Bethesda, Maryland. And in his presentation in 1964, he described the original methods utilizing collagenase, a crude preparation of collagenase, for the isolation of individual fat cells, as well as what he called the vascular stromal fraction. And it was during his Nobel lecture in December of 1994, and also a follow-up autobiography that he wrote, that he described his use of collagenase in order to digest fatty tissue and then isolate individual cells. He also noted in his autobiography that this collagenase was actually an impure preparation that contained many proteases. He had tried other proteases in the past that were unsuccessful for the isolation of fat cells, but it was this impure collagenase, a collagenase that he obtained through the Worthington Corporation, that really gave him the optimum isolation that he sought. I've shown in this slide his presentation, or his method for the isolation of individual cell populations from fat. He used rat tissue as his source of tissue. He minced the tissue into 3 individual pieces, so it wasn't heavily minced. The enzyme that he used was Worthington collagenase. At that time, this was, again, an impure collagenase. It was the 40% ammonium sulfate cut of the Clostridial digest, or Clostridial enzyme preparation. He placed the fat and the enzyme in a 25 milliliter Erlenmeyer and then agitated it constantly in a shaking water bath for 60 minutes. He then took the digest, centrifuged it at 200 times G for 10 minutes, and what he found was, and you can see in the center portion of this slide, an example of the centrifuge resulting pellet, as well as the cake of cells that forms. The top cake of cells, as we call it, are the adipocytes. They're buoyant, free-floating. There's a intermediate supernatant that contains media, collagenase, as well as a number of free cells that don't have the density to pellet at this particular centrifugal rate. And then the pellet of cells, which he defined in 1964 as stromal vascular cells, as well as a pellet of RBCs at the bottom of this centrifuge tube. And these are his original images. In A above right, these are the free adipocytes that he went on to study and received his Nobel Prize for these studies of these cells. And then the stromal vascular cells, this is a phase contrast micrograph in his original paper that showed the microvascular fragments that he obtained and defined as the stromal vascular cells in the pellet. Now, what I've done during my studies is, starting with 1964, if you do a PubMed search utilizing the terms adipose, cell, and isolation, you'll get over 13,000 hits, 13,000 publications that utilize these three terms. I have studied a large number of these 13,000 publications to evaluate all the different methods that have been used to isolate cells from adipose or fat tissue. It's called by either term. And the first hits were found in 1964, Rodbell studies, and you can see over the years there has been an increase in the number of publications utilizing adipose, cell, and isolation. Now, what I've done in reviewing these papers is I've come up with somewhat of a unifying terminology and the basic methods that have evolved for the isolation of cells from fat, starting with Marty Rodbell's studies in 1964. One of the next procedures that was published in this series of procedures was work performed by Dr. Roger Wagner in the late 1960s, early 1970s. In his publication in 1972, he used the Rodbell method utilizing rat fat, minimal mincing, the Worthington enzyme, a 50 milliliter Erlenmeyer, shaking water bath for 45 minutes. He centrifuged this digest, got the fat cells that went to the top, threw those out, and then what he did was took the resulting pellet and the supernatant, and he passed it through a 200 micron stainless steel filter, and then obtained the cells that were captured on the top of this filter and evaluated what those cells looked like. And this is figure 1 from his publication, showing the microvascular fragments that were formed. In this slide, I'm showing you his hand drawing of this procedure from 1972. The fat cake of cells that results following collagenase digestion, the supernatant, and then the pellet of cells, which he again called, as Rodbell called it, the vascular stromal pellet. They're vascular stromal cells. He then took this pellet and supernatant, passed it over the 200 micron filter, and what filtered through were the free stromal cells, and the capillary tufts, the microvascular fragments, were held up on the filter, washed those off, and then Roger went on to do studies evaluating these cells as well as culturing these cells, and we'll talk about that in some future presentations. But this really was the second method developed for the isolation of cells from fat, where Roger Wagner isolated and identified the microvascular cells that exist in the pellet of this digest after you pass it through a filter. A third paper that was reported by an investigator by the name of Per Bjorntorp. And what Dr. Bjorntorp did in 1978 is published again utilizing the Rodbell method for taking fat from a rat, in this case epididymal, just like Roger Wagner, minimal mincing, Worthington digestion, an Erlenmeyer water bath, and this was 30 minutes of digestion. And then he used 2 filters to remove the microvascular endothelial cell component from this digest. And the cells that pass through these filters, a 250 micron and then a 25 micron filter, he took this solution containing the cells, centrifuged it, and then defined the final product that came out of this process as the stromal cell fraction. You see his original picture of this stromal cell fraction that has been put into culture. So this is the primary isolate and put into a culture. He did not publish what the initial cell fraction looked like. He only published what the cultured cells looked like. But he stated in this paper that the cell aggregates, consisting mainly of endothelial cells, were separated from this initial digest, and then he pelleted the filtered cells and called them the stromal cell fraction. Now, I want to point out at this time the differences between Rodbell, Wagner, and Bjorntorp, and say that in Rodbell's procedure, no filters are used. In the Wagner procedure, the filter is used and the microvascular fragments are isolated. And in the Bjorntorp method, a filter is used to remove the endothelial cell population, giving you the stromal cell component. Now, this is critical because the Bjorntorp method has been used by, in my review of the literature, a majority of investigators who study cells isolated from fat, not the buoyant adipocytes, but those more dense cells that can be pelleted by centrifugation. But Bjorntorp's method does not result in a stromal vascular fraction. His method results in a stromal cell fraction and has removed the endothelial cell population. Now, what has happened since, again, the earliest reports by Rodbell of the 1964 method for the isolation of the stromal vascular cell population, is numerous investigators have used either the Rodbell method, the Wagner method, or the Bjorntorp method to get a pellet of cells that they have generally called the stromal vascular fraction. But they've essentially renamed this fraction utilizing a variety of terms. I'll go through this quickly, but you can go back and pause on this slide in the presentation and take a look at how these names have emerged. Rodbell, 1964, the stromal vascular cell population. Ing and Poznansky, same essential isolation procedure of Rodbell with or without filters, and they call it sedimented cells. Wagner in 72, he called it capillaries or capillary endothelium. Poznansky, stromal cells. Van was the first one to call it the stromal vascular fraction in 76. Bjorntorp, using the exact same method that he had published earlier, he called these cells the pre-adipocytes and the adipoblast. He was very interested in his career, he studied obesity, and he wanted to understand what the pre-adipocytes were. He called the stromal vascular fraction pre-adipocytes. Work from my laboratory, I called these cells the microvascular endothelial cells, and I'll go through the rest without naming the investigators. Adipocyte precursor cells, human adipose endothelial cells, brown fat precursor cells, microvessel endothelial cells, human omental microvascular endothelial cells, adipose tissue-derived stromal cells, stromal vascular fraction process lipoaspirate cells, or PLA, adipose tissue-derived stem cells, or stromal cell pre-adipocytes. In all of these presentations, publications, all of these investigators did initially the Rodbell method, but then they used differing variations on the technique, which included different types of collagenase times, as well as different filtering techniques to get their final cell population. They described that final cell population initially as the vascular stromal or stromal vascular pellet, but then many of them just renamed it as all the different names that you see here. And what I've done in this slide is taken that original series of drawings of all these different methods and put them all together so you can compare the methods for the isolation of cells from fat with the different names. And then I've also given you some of the most important CD markers to describe these cells. Rodbell, enzymes, agitation, centrifugation, pellet, the stromal vascular fraction results, and these are CD29 positive, CD31 positive, and CD45 positive. It is a heterogeneous mixture of cells. Wagner used the Rod-Bell initial procedure, but then he captured, using this 200 micron filter, the microvascular fraction. These were CD29 positive, CD31 positive, and CD45 negative cell populations. This was the somewhat pure endothelial cell population from fat. And then Bjorntorp, utilizing what I consider one of the most commonly used methods today for the isolation of what people often call the stromal vascular fraction. He used the Rod-Bell method, and then he used two filters in succession, a 250 micron filter, a 25 micron filter, centrifuged the material that came through the filter, removing most of the endothelial cells, and he ends up with a stromal cell fraction. Some investigators will call this a stromal vascular fraction. I believe that is incorrect. This is the stromal cell fraction. It is CD29 positive cells, not all of them, but many of them. They're CD31 negative, but they're also CD45 positive. And then the non-enzymatic methods of Carrel, initially in 1923, that have become very popular today, the non-enzymatic isolation of cells from fat. The fat is minced, it's agitated, and centrifuged, and you end up with a fraction that some again will call the stromal vascular fraction. It is not. The endothelial cells in this case are not released from the fat, and all you're getting is the stromal cell fraction, and that is the correct terminology. They're CD29 positive often. They're CD31 negative most of the time because the endothelial cells are not released without the enzymes, and they're CD45 positive. So I provide to you now somewhat of a unified terminology to describe the cell populations that can be isolated using these four basic methods. And then I also provide a recommendation that everyone who uses these different four methods, when they are providing a primary reference to the methodology, they cite one of these four authors. The first one is Enzymatic Digestion Utilizing Either a Crude or a Purified Collagenase plus Protease greater than 20 minutes at 37 degrees, and then taking the digest and either not using a filter or using a filter that has at least 200 micron pores in it. The resulting pellet of cells is what I consider the adipose stromal vascular fraction cell population. This digestion and the enzymes are critical to break the microvascular cells out of their extracellular matrix, and then also to break these cells down into smaller fragments that will pass through this 200 micron. If you use this method and you're isolating SVF, you really should use the Rod-Bell reference as the primary citation. And I also, and I'll show in this next slide coming up, believe that you should provide all of the methods that you utilized for the isolation of cells and not simply say it was done according to the methods of Rod-Bell, because there have been so many changes in some of the methods that have been used. But this is the SVF fraction. The Bjorntorp method is enzymatic digestion. It is often less than 20 minutes, but the timing is not as important as the filter that is used. For those investigators who use a filter less than, I'm choosing 200 microns as that separation point. If you're using a filter less than 200 microns, the cells that you get that pass through this, I consider the adipose stromal cells. Of interest, many investigators will send an isolate to the fluorescence laboratory, the FACS laboratory, and very often they will use a 70 or a 40 or sometimes a 20 micron filter before they'll put the cells through a FACS analysis. Most of the endothelial cells are removed before that FACS analysis. These are adipose stromal cells, and the primary reference should be given as Bjorntorp. If If you're using enzymatic digestion collagenase and a lower protease, less than 20 minutes, and you capture the cells on a filter and then utilize or look at the captured cells, these are adipose microvascular fragments, or MVF. And the primary reference should always be Wagner for this. He was the first one to really describe this methodology. And then non-enzymatic tissue disruption. These are adipose stromal cells. The cells are not vascular to any great extent. The vascular fraction does not come out without the use of the enzymes. And the primary author on this really is Carrel from 1923. Of interest, two of these investigators, Rodbell and Carrel, got the Nobel Prize for work that they performed, and then Bjorntorp and Wagner really should be cited for these methods. Now, I'm also providing this slide. It will come up, and I'll talk about it in much more detail in subsequent presentations. But these are the descriptions that should be provided in publications, presentations, patent applications to fully describe the methods that are used for the digestion and then the subsequent isolation of cells. I'll have presentations on some of the individual components of this, including techniques for liposuction and mincing of tissue, a presentation on the enzymes that can be used for digestion. And then again, you see as this passes through different filters that are used. You capture the cells on top. These are the fragments. The cells that come through, depending upon the filter size, are either the stromal vascular fraction or the stromal cell fraction. And then subsequently characterization of these cells. There are numerous methods that are used to characterize these cells. I'll also talk in a later presentation, but I thought it important to bring up here, that fat as it is removed from the body has a certain population of cells, and there are only two publications that I've been able to find. Hopefully there are more, and you'll let me know of these by utilizing my email and sending me papers that I may have missed. But there are only two publications that I found where investigators have taken a piece of fat in situ, have sectioned it, and then evaluated the cellular population. And what you find is in whole human fat, about 12% of the cells are adipocytes. Over 60% of the cells, before any isolation, before anything is done, over 60% of the cells are endothelium. Paravascular cells, 2% or less, and the rest of the cells are formed element cells, leukocytes, macrophages, T regulatory cells, and monocytes. So this is your starting material. If you look at the cells that you get after an enzymatic digestion, this is from the published literature that I've reviewed up to 2025. The distribution, relative distribution of cells in isolated stromal vascular fraction, but it's also stromal cell fraction and the microvascular fraction. People will often call this just the stromal vascular fraction. And you see the variation in cells. Some isolations, you don't get any endothelial cells at all, 0%. Well, that's probably because you're using the Bjorntorp method and you're filtering all the endothelial cells out, or your markers for endothelial cells are not the true characteristic markers that need to be used. You'll see a variety of variations, huge variability in the relative percentage of cells, but you have to remember you're starting with more than 60% of these cells being vascular endothelial cells. Where do they go? Some are captured on filters, some are not even released, the non-enzymatic methods. And I leave you with this slide for this presentation. This is a phase contrast image of what I'll call a more modern stromal vascular fraction isolation, where a somewhat quote-unquote optimized collagenase digestion is used and a filter that is 200 microns is used to filter out large clumps of undigested tissue. These microvascular fragments that you see here do go through, and there are lots of individual cells that are here. In this type of isolation, when you do the characterization of endothelial cells, more than 50% of the cells in the stromal vascular fraction are endothelial cells. So thank you for listening to this. Here's how to get in touch with me. My email address, cvregen@gmail.com. I've also provided the URL for my website, cvregen.com. And if you go there, you can find the series of presentations on adipose technology. Thank you.